Welcome back, Agriscience. Today we're going to look at biotechnology and what it has to do with the plant industry. So we've had a little bit of biotechnology with several different areas this semester. We've talked a little bit about biotechnology in animal science. We talked a little bit about biotechnology with environmental science, but biotechnology within the plant industry is probably more important than any of the other areas. And the reason is because there are so many more ways to apply biotechnology to the plant industry. So let's kind of talk about some of the most important ways that we do that. First, let's talk about what biotechnology is. And basically, we're using living organisms to make new products or to make plants do different things or change the way they process things or solve problems. For example, a new product would be yogurt, and yogurt is not necessarily something, some Frankenstein monster thing that's created in a lab, but basically you're using yeast to help to ferment that milk product in order to turn it into yogurt. Tissue culture is a new process, and it's actually been around for quite a bit, so it's really not a new process, as far as recently, but it's been around for quite a bit. And tissue culture is where you're actually taking tiny little pieces of sterile plants, and you're putting it into a petri dish that has a nutrient solution on it, and you're producing new plants in a lab type setting. Not something that we can really do very well here, but we will talk a little bit more about tissue culture as we go throughout the slides here. As far as throughout history and some of the first things that ever happened with biotechnology, yeast was used to make bread rise. And so prior to the use of yeast, a lot of breads were flat. And so you really didn't have what you would have heard of as unleavened bread. So there really were not those nice fluffy things like you would have donuts and yeast rolls and things like that. Bacteria is used to make cheeses. Microorganisms and other bacteria are used to transform fruit and grain into alcohol. You've got bacteria that produces silage. And we mentioned silage a little bit whenever we were doing the animal nutrition portion. Silage is just basically a fermented, usually green corn stalks. And then E. coli, which we usually think about as being a bad thing, but there are many, many strains of E. coli, and there's only one that's really dangerous, is the E. coli 0157H7. That's the one that comes from cattle and other ruminant animals that can cause illness. But most E. coli, every animal sheds E. coli of some sort, including humans. And so there are some E. coli bacteria that are actually used to produce insulin, which helps those people who have issues with diabetes. There's just a picture of yeast during its growth stage, and a picture of silage. And we've already mentioned silage a time or two in the course. So as far as kind of the history and the basics of it, you probably can remember, and you probably have heard this in some of your other classes, Gregor Mendel was actually a monk, and he was, more or less in this monastery, and his hobby project work that he did was actually crossbreeding pea plants. And he figured out that he could cross wrinkled peas with smooth peas, and he would get the same results most of the time. And he could cross peas that were tall versus short, and ones that had purple flowers versus ones that had white flowers, and they would get different results every time. And so he kind of figured out that parent organisms pass on genetics to their offspring. And so once he figured that out, then we were able to start looking at heredity based on animals and humans and all different types of things, and we could start to understand a little bit more about genetic material. I really wish that we could do Shockwave on these Chromebooks, because I have a really great game that is a lot of fun to do with Mendel's peas, and you cross the peas and they come up. But unfortunately, technology is what it is. Heredity just means the transition of characteristics from one organism to its offspring. The genes are what helps to determine that, and the these are the characteristics that are found within DNA. And then the term generation just means the offspring of one particular set of parents. DNA is where everything comes from as far as what makes you you. It's your DNA, and DNA is wound into what we call a double helix. It basically looks like if you took a ladder and you held it from the top and the bottom and you twisted in opposite directions. That would be what a double helix looks like. It just holds all the genetic information that a cell would have. And there are different places within DNA that contain certain genes, and those genes are what help to determine certain traits and characteristics about you. And so DNA is more or less what makes you you and what makes you different from the students around you, but what also makes you different from, a pine tree, or what also makes you different from a alpaca. So it's what makes us into different types of organisms. So the gene is a small section of DNA. Usually that codes for one thing or another. Like it may be a gene for eye color, it may be a gene for hair color, it may be a gene for your susceptibility to certain types of diseases and cancers. So genes are all over your DNA, and they all have got different pieces and parts of the puzzle. Now, in order to figure out how those pieces and parts of the puzzle work, scientists have done what's called gene mapping, and they've figured out where certain genes are in your genetics. And that gene mapping helps us to be able to look at a person's genetics and figure out, what is the possibility of this person having this illness? What is the possibility of this person passing on this particular trait to their child? And so gene mapping helps us to be able to do that. With DNA, you have 4 different bases, and the 4 different bases are adenine, thymine, guanine, and cytosine, and they always only pair with their partner. So A and T only pair together. You will never see A pair with C or G. They only partner with the one that they're supposed to partner with. Guanine and cytosine only partner with each other. And so as you're doing DNA, it's kind of, as you're looking at DNA, the order of how they are on the ladder, like whether the A is on the left rung of the ladder or the T is on the right rung of the ladder, or vice versa. It's what makes you you. And so the coding and the sequence and the way these bases are put together are what makes you different from other human beings. So in the early 1980s, we started doing this thing called genetic engineering. We had always used biotechnology to a certain extent with the bacterias and the microorganisms that were used in order to create new products. But we started engineering the DNA of things. And basically what I mean by that is you can literally move genetic material from one organism into another organism. They do that through what's called gene splicing or recombinant DNA technology. You can make a plant less susceptible to disease. You can make a plant resistant to insects. You can make a plant resistant to herbicides. For example, if you've ever heard of the herbicide Roundup, Roundup will kill pretty much any plant that you spray it on. However, genetic engineering was done in order to create what we call Roundup ready crops. And these are crops that you plant out in the field. And then once you plant them out in the field, they have been genetically altered to not be susceptible to Roundup. And so when you spray that field, maybe you have Roundup ready soybeans out in your field. You spray that field, it only kills the weeds. It will not kill your Roundup ready soybeans. And so There are so many new things that are being invented as we speak. There are so many things that are in the process of being tested and researched, and so genetic engineering, big important part of agricultural production these days. Cloning is, we talked a little bit about cloning with animal science, but cloning is much different in plants than it is in um, animals. And with cloning, you basically are creating an exact duplicate of another organism. And there are several ways to clone. This one says micropropagation, which is the same thing as tissue culture. And tissue culture, I mentioned a little bit ago, and that's where you're actually taking those sterile pieces, putting them into a petri dish, putting them under a grow light, and they actually grow in a petri dish, and then they get transferred over to another sterile environment. Um, Taking cuttings, which we're going to learn about next week, that's also a way to clone plants because you are literally taking identical genetic material from the parent plant and you are giving that genetic material to new babies. So it's just another way of doing cloning. Um, Sometimes, as far as biotechnology, we have what's called an indicator species, and I think we talked about that a little bit before too. With indicator species, you're using certain plants, animals, microbes, insects in order to warn you about pollutions or pollutants that are in a certain area. And if you have a certain type of moss in a certain area, then you know that you have particular issues with one thing or another. It same thing works in ponds. Certain insects will only be found around and in certain ponds, and so those indicator species help us to be able to figure out what's going on in a particular area. Bioremediation is using living organisms in order to clean up toxic waste in water and soil. Sometimes we do this with oil spills. There have been times in the past where you've had issues with oil that has been released into usually salt water, and it affects those animals that live in the salt water. And so there is actually a bacteria that can feed off of the oil to help clean up the um, issues that we have. It can help clean up the oil. Biostimulation is adding nutrients in order to stimulate the growth of microbes that are going to be beneficial. So it just makes things work more quickly and work more efficiently. Um, Phytoremediation. This is kind of what we got going on a little bit with the aquaponics system that we have in the greenhouse. So the aquaponics system in the greenhouse is we have catfish in there if they don't quit jumping out of the tank. We have catfish in there, and as they release waste in the water, that water is going to be fed into the aquaponics bed, and it will be used to help fertilize the plants that are in there. And then the clean water will be released back into the tank for the fish to be able to swim in. So it's basically like a filtering process. Animal reproduction in production, we talked about biotechnology. Whenever we did that unit, several different applications there. Some fuels are actually created from plant material, ethanol. You actually can create fuels out of wasted French fry oil. You can create fuels out of bacteria. You can create fuels out of potatoes. So there are a lot of different types of biofuels that could be an alternative to using oil. Um, these things, whenever we recombine this DNA, and basically you're just taking DNA and you're taking it from one organism, putting it into another. They don't even have to be related types of organisms. You're doing it just to improve the plant's performance. In order to make it something that's more useful, more beneficial to the consumer. It might help to control disease or insects or weeds or something like that. But it's the purpose of it is to make something work better. There are some concerns, though. There are a lot of people who have concerns about the idea of using biotechnology. Is the government monitoring things enough? Are there issues with allergies in consumers? So is it safe for consumers to be able to use? Is it safe for the environment? Do we even understand what we're doing? Have we labeled things properly? Some people don't want to look at the labels. Some people don't want to use GMOs. Some people are very strict about looking at the labels. Usually, if you look at a label and you see something that says modified, then that is a GMO. You probably are using some sort of Roundup Ready crop. But it's not anything that should hurt anybody, but it is a means for being able to produce more product. There are some people that think of it as an ethical issue. What's right, what's wrong. Are you okay with using biotechnology as far as plants? Because, the plants are not, I mean, of course, they're a living organism, but they're not going to feel the same way as when you use biotechnology with animals. So what is right? What's wrong? What's crossing the line? What can you tolerate versus what are you going, I'm not so sure about. This just talks about the steps of the scientific method. This is the same scientific method that you have used throughout your student careers. If you're trying to solve a scientific problem, you want to identify what that problem is first. You want to do some research on it, form a hypothesis, and that hypothesis is just an educated guess. You're going to prepare your proposal and design your experiment, then you're actually going to do the experiment. Once you do the experiment, while you're doing the experiment, you're going to collect data. Once you've collected that data, you're going to come up with your own conclusions. And typically you'll write some sort of a written report. We do have opportunities for students to do Agriscience fair, which is basically like doing a big science fair project that's related to agriculture. And we had a student that did that last year and went on to national competition. So the opportunities are out there if that's something that you are interested in. As far as biotechnology and the plant science industry itself, you have herbicide and insect resistant crops. I've already mentioned the Roundup Ready. It contains a gene that just does not make that plant susceptible to Roundup. There is another one, though, that we call BT, and Bacillus thuringiensis is an organism that actually lives in the soil, so it's safe. It is not going to hurt animals, or it's not going to hurt people. It hurts one type of animal, but it's not going to hurt people. But it's an organism that insects don't tolerate very well. And so these BT crops have been genetically modified to contain BT. And when insects eat the plant, it kills the insects. And so it's basically a natural type of insecticide, but it's not dangerous to humans. We have created crops that have got better nutrition, longer shelf lives. One of the very first, and you can read about the peanut and the sunflower, and basically it's just producing oleic acid and producing more of it than what they normally would. But one of the very first types of genetically modified crops was a tomato, and it was called flavor saver tomato. And these flavor saver tomatoes were specifically designed to last longer in the grocery stores. And if you notice, when you go to the grocery store, a lot of things don't have the same shelf life as what they used to have. They have a much longer shelf life. So we have done a much better job of keeping things fresher longer so that they can have time to be picked and processed, shipped, and put into the grocery store and still be a quality product for the consumer. This is tissue culture again. This is that micropropagation. You can actually start them in a petri dish, and once they get a little bit bigger, you actually just take a very tiny piece of plant and put them into a petri dish. It does have to be done in a sterile environment. We've tried to do it at the high school before, but high schools aren't very sterile, and so we have not had tremendous amount of success with that. But you start it in a petri dish. Once they begin to grow, then you can move them over to like a test tube, like they're doing here, and have time for it to get a little bit longer. The nutrients that are down here in this auger are helping that plant to be able to get what it needs in order to be able to grow, and then eventually you would be able to transplant it out into a pot. And that is it for biotechnology. And so your assignment for this week, you're going to be looking at creating some sort of a biotechnology type plant, something that would be useful on the market for people who are looking for uh, you know, super plants. So if you have any questions about that, make sure that you get in touch with me, and I will see you guys next